Hello, in this video we will discuss about the plasma membrane. So the plasma membrane we will discuss about the fluid mosaic model. So the first of all I want to share that the content of the plasma membrane. So the plasma membrane contains 60 to 80% protein while the phospholipids or lipids contain 20 to 40% and the cholesterol in small amount as well as carbohydrate in small amount also. So, in this way, the S. Jonathan Singer and Garth Nicholson, which that is the two scientists create or discover the plasma membrane structure, and they give the name is known as the fluid mosaic model of membrane, and this fluid mosaic model of membrane structure in 1972. According to this model, a membrane is a double layer of proteins as well as phospholipids. It's present in fluid form, so it is in the form of fluid form, and it is the protein embedded into the phospholipid bilayer of the phospholipid, so you can understand as well. So the first of all we will understand about the unit of the plasma membrane. So contain a hydrophilic and hydrophobic interaction. In this way the phospholipids phosphate group is used for the interaction of the hydrophilic, and it is the polar head and absorb the water, while the inside the hydrophobic non-polar tail, which that is basically the non-polar tail, and it is used for it. And in this way here is you can see that is the cholesterol. This cholesterol is basically used for the a hindrance or prevention of the water inside to move, so that maintain the homeostasis of the water inside and outside through a cholesterol. And cholesterol is also stabilize membrane and less water soluble and maintain the fluidity inside. So inside and outside the fluidity will be maintained, as well as the stretching of the plasma membrane for the turgidity or increase of the area of the cell membrane is important. So let's begin to understand about the fluid mosaic structure is here. This is the structure look like this, contain a bilayer of phospholipid, and in this way here is the hydrophobic and of phospholipids. In this way the hydrophobic inside, so this is the phospholipids, and basically here is the hydrophilic interaction. In hydrophilic is the outer phosphate group which that used for the hydrophilic, it means the water loving, while the inside the water dreading or water fearing due to the fatty acid. Fatty acid is contain a unsaturated and saturated fatty acid chain. So here you can see this is the peripheral protein, and here is the alpha helix protein, which does contain a carbohydrate. So the oligosaccharide is the side chain will be occur, and here is the peripheral protein, which that is also attached with the carbohydrates known as the glycoprotein. So here is the globular transmembrane protein. It is also known as the intrinsic protein. So here is black color is showing in the cholesterol, cholesterol dispersion or cholesterol in presence. So here is the globular transmembrane protein are present here, and here is the alpha helix protein, and in this way the glycoprotein formation and glycolipids formation due to the glucose with attached with the lipid. So let's begin to understand with the respect with the carbohydrate attachment. So when the carbohydrate attach with the proteins known as glycoprotein, and when bind with the lipids known as glycolipids, when bind with the lipids, protein and carbohydrate collectively is known as the glycocalyx. Glycocalyx show molecular fingerprinting as well as control the behavior of cell. In this way the behavior of cell is due to the production of receptor and receiving the different type of the ligand on the different receptor due to the conformation change through a glycocalyx formation. So in this way you can understand as well with a better way to understand you can. And here is the cell coat. The cell coat is basically the carbohydrates and protein and lipids. Cell coat contain carbohydrates, protein and lipids. And in this way the cell coat will increase, it makes the receptor on cell. So the cell coat is contain also the ligand binding receptor, for example epinephrine, glucagon, insulin, etc., which that is give the response of the cell in the different tissue of the human body and different organ. So let's begin to understand important function of cell membrane first of all. So the first of all we will discuss about the materials movement. It is in the form of three types: the endocytosis, exocytosis and transports. So basically the endocytosis and exocytosis, first of all we will understand about the material movements. So this is the three type I am drawing here: that endocytosis, exocytosis and transports. So endo mean inside and exo mean outside. So the endocytosis is in two type and exocytosis also. Phagocytosis and pinocytosis. Phagocytosis means the solid particle will move inside, while the pinocytosis solid particle move in or out to the cell is known as the liquid particle. So here is the transport is in type active transport and passive transport. Active transport use energy and active the against the osmotic gradients. For example, the nerve impulse conduction, so the coordination in the nervous system. While here the passive transport is different type. For example, the osmosis, diffusion, for example simple diffusion, facilitate diffusion, and the osmosis is the semi-permeability, due to the semi-permeability the water movement, while the diffusion is direct gases diffuse into the cell and out to the cell. So the facilitate diffusion is through channels, and that is the channel basically, so channels of protein. So let's begin to understand. The second one is the separate the inside of the cell from outside of the different cell. So this is the separation barrier. So here is the receptor are also present in the plasma membrane. In this way the G coupled protein receptor, ion linked channel receptor and enzyme linked receptor. For example, G coupled protein receptor contain a G protein inside. So the ligand will be bound here, you can see, and the GPCR is here and the G protein is here. So the cyclic AMP will be trigger the active transport. While here you can see the ion ligand. In this way the ion ligand will be bound and ion will move inside, and due to the ligand will be bound as an ion, and that is the material move inside due to the opening of the gate of the plasma membrane, the protein gated channel. So in this way you can see the open and move the material through a ion ligand, while the enzyme linked receptor use the enzyme for the opening of the gate. So thanks for watching. Please make sure to subscribe, like and share this video.